Hey guys, so we are now going to start with the next part of this lecture. So we've just finished the mouth, talking about the tongue and the teeth, and now we're going to go into the pharynx and the esophagus. So after we swallow, basically it's going to go from the mouth into the oropharynx, into the laryngeal pharynx. And then once it goes through that area, it's going to go into the esophagus next. Now, the esophagus is basically a flat muscular tube. And it basically goes from that laryngeal pharynx to the stomach. This guy is basically collapsed when it's not involved in food propulsion. It'll go through the diaphragm at an area that's called the esophageal hiatus. And basically, it's going to connect to the stomach at the cardial orifice. Let me go up here. Now, when we look at this, the esophagus does have all four alimentary canal layers. And basically, you have within the esophagus glands called esophageal glands. They're located in the submucosa, and they help to secrete mucus to aid in the movement of the bolus. And again, if you remember, there is the area that's called the muscularis externa, and this has skeletal muscles basically that help to mix, and then smooth muscles in there as well. And so that helps with basically the movement or the propulsion with the peristalsis and the mixing that happens less likely in the sedimentation. Yeah. Great. I'll move forward. Sorry about that. So here, let's look at the layers of the esophagus here. You can see the lumen in the middle, and then on that interior is the mucosa that is in that area. And basically, just to let you know, there's stratified squamous epithelial, and it does change once it hits the stomach to simple columnar. Not truly important to know, right? Then we're going to have the submucosa, and this is where the basically mucus gets secreted in this area, and that's going to help to basically move the bolus within the esophagus. Then, of course, you have the muscularis externa. And this is what helps with the movement of the bolus throughout. And here, instead of the serosa, you have that inventita. And basically, this is just more robust. And so those kind of give you the different layers of the esophagus. So, the pharynx and the esophagus are conduits to pass food from the mouth to the stomach, right? And basically, the major function of both organs is propulsion, and that's essentially to get the food that becomes a bolus in the mouth through from the mouth to the stomach. And that starts with deglutition, and that's the process of swallowing. This involves the coordination of about 22 muscle groups and 2 phases, the buccal phase and the pharyngeal esophageal phase. And so the buccal phase is basically the voluntary contraction of the tongue. And basically the pharyngeal esophageal phase is an involuntary phase. This mostly involves the vagus nerve. And it's basically controlled by swallowing centers. And those are located both in the medulla. Something's wrong with your medulla oblongata. And the lower pons. So let's talk about that process of swallowing.
The oropharyngeal and esophageal stages. At the start of a swallow, a food bolus is voluntarily pressed by the tongue up against the roof of the mouth and backwards towards the pharynx. In response to activation of pharyngeal pressure receptors, the swallowing center in the medulla initiates reflexes that prevent food entry into respiratory passageways. The uvula contracts, which blocks the nasal passages from the pharynx. The laryngeal muscles contract, closing the glottis at the top of the trachea by tightly aligning the vocal folds. The epiglottis swings down upon the closed glottis. With all airways blocked off, respiration is temporarily inhibited. As the upper esophageal sphincter relaxes, pharyngeal contractions drive the bolus into the esophagus. The oropharyngeal stage is done, and breathing resumes. During the esophageal stage, a primary wave of peristalsis initiated by the swallowing center pushes the bolus through the esophagus. As the bolus travels through the esophagus, the lower esophageal sphincter relaxes, allowing the food to enter the stomach. If the bolus is sticky and adheres to the esophagus, secondary peristaltic waves triggered by the intrinsic plexus at the point of distension completely clear the esophageal lumen to finish the swallow.
And so if you look at this, guys, this area, let me give a different color here. This area that's right here, where the basically the esophagus joins the stomach, that's what's known as the cardinal orifice. And in there, that closes it off, right? That keeps this opening or this orifice closed is the gastroesophageal sphincter. It's also known as the cardiac sphincter. So this surrounds the sphincter, surrounds the cardinal orifice, and it keeps the orifice closed when food is not being swallowed. Mucus cells on both sides of that sphincter help protect the esophagus from acid reflux. Okay? And so, if you look at here, you have two phases, right? You have the buccal phase, which once you allow food here, right, the first thing you're going to do is it's going to have this upper esophageal sphincter is contracted or closed, and you can see that right in that area that I've pointed to. The tongue will press against the hard palate and force the food bolus into the orthopharynx. And so the orthopharynx area is that back area right there. So that's going to be the buccal phase that happens first. And the pharyngeal esophageal phase begins once the tongue blocks the mouth. You can see that there. The soft palate and the uvula will rise, closing off the nasopharynx, which is that area at the top, right? So that it can't go up into your sinus cavities. The larynx rises, so the epiglottis blocks the trachea, and so you can see that in that area right here. And the upper esophageal sphincter relaxes, which was originally right in this area, right? And allows the food to actually enter the esophagus. And you can see it'll go down there. And as this phase continues, basically the pharynx will contract, forcing food into the esophagus as it goes down. And that upper esophageal sphincter will contract again once the food starts its way down the esophagus. From here, peristalsis will move the food through the esophagus to the stomach. And you can see at this point that the esophageal sphincter is closed. That's also known as the cardiac sphincter, right? And basically the food or the circular muscles will contract, pushing the food through that sphincter, and it'll open. And that sphincter closes around the cardiac orifice, right? So that orifice opens. And then after the food enters the stomach, the sphincter will close, preventing regurgitation or all those juices coming into your esophagus. And so I thought this was kind of cool if you wanted to see what it looked like with someone actually swallowing. So here there, you can see the food moving around from the stomach. And see all of this. And you can see some of that going from the buccal phase, the very beginning, and then once it starts moving down, going into that pharyngeal. What's there? You can see everything moving there.
Recording.
So that kind of gave you an idea of what it looked like. Now the next part that we're going to talk about is once it gets through that orifice into the stomach, what happens. So the stomach is the temporary storage tank, and this is where much of the chemical breakdown of protein starts, or protein digestions. Carbohydrate digestion started in the mouth with the amylase that you have. And so basically what's happening here is we're going from that bolus, and the stomach is producing chyme. An empty stomach is somewhere about 50 milliliter volume, but it can expand up to 4 liters when you're basically eating. And as you can see these little lines in here, and they are called root. And basically these are the stomach folds, right? They help to increase that surface area. And so let's look at it and get a general area. So you can see this is the esophagus. So that opening right here is the cardinal orifice. So you can see this area towards the top is called the cardia. And so this area is what actually surrounds the cardial orifice. Now, if you look at this dome part at the top, this is going to be right beneath the diaphragm. Diaphragm is up here, right? That's going to be what's known as the fundus. Now, the midpoint of the stomach is the body. And then you can see at the end is the pyloric area. This is wider and more superior part of the pyloric region. So you can see the pyloric atrium, the pyloric canal here, and then the pyloric sphincter over here. So basically it starts with the superior region, which is the antrium here, and then it'll go through, and basically the canal gets even kind of more narrow, and then it terminates in the pylorus, specifically this region that's called the pyloric sphincter at the pylorus. And so this is what closes, right? And this pyloric valve or sphincter controls the emptying of the stomach into the small intestines. That area is specifically called the duodenum or duodenum. Now the layers are similar here, and you can see that in the stomach you have the circular layer, the oblique, and the longitudinal layer of all these different basic muscles in here, right? And so as we're going through here, you can even see they're talking about the greater curvature area that's down here in this, right? This is that kind of convex lateral surface of the stomach. The lesser curvature is on the other side. As you can see, that curve is a little bit kind of smaller, and this runs from basically the lesser, well, this is the concave medial surface of the stomach. The mesenteries extend from this curvature. So this is, you're going to get the mesenteries extending from this side, right? And tethering the stomach to other digestive organs. I have it on here. At this point, we're going to, you can go the omentum. There's the lesser and greater omentum. So the omentum, or specifically the lesser omentum, is going to basically run from the lesser curvature to the liver. And the greater omentum is going to drape from the greater curvature, right? And basically this is going to drape over the intestines, the spleen, and the transverse colon. So that's basically the anatomy of the stomach.
What exactly happens in our stomachs when we eat? Even before we have taken the first bite of a meal, the brain sends impulses to the stomach. These impulses start the production of secretions in the stomach, and the upper part of the stomach, which acts as a reservoir, expands. The food passes into the stomach through the cardia at the stomach entrance. This is a muscle that acts like a valve, closing the top of the stomach. The top part of the stomach, the fundus, is where food and the air that we swallow with every bite is collected. This is where the stomach volume starts to adapt. The fundus is an extremely adaptable structure. The more food goes into the stomach, the more it actively expands. In the middle part of the stomach, called the body, gastric juice is produced and mixed with the mass of chewed food. The main component of gastric juice is gastric acid. This eliminates bacteria in the food and also helps to prepare the food for the following stages of digestion. The muscular contractions of the stomach wall churn the food mass and mix it with gastric juice. 3 contractions per minute move the food mass back and forth. After a certain time, the contents of the stomach are adequately broken down and mixed, and are passed in small quantities into the intestine via the pylorus. As the stomach empties, it gradually returns to its previous size. Fair.
So that kind of gives you an idea of how the stomach works and what we're going to be talking about for this time. So the control and the blood supply of the stomach. The autonomic nervous system supplies the stomach. So you have sympathetic fibers that originate from the splenic nerve in the thorax that are actually relayed through the celiac plexus to help with supplying the control of the stomach. Parasympathetic fibers are supplied by the vagus nerve. If we're going to start or stop digestion, right? And the blood supply that comes from the stomach essentially is the celiac trunk. That's the gastric and splenic branches. And then, blood that leaves from there goes from the veins of the hepatic portal system, because again, we're going to want to clean that blood before we take it and put it back into circulation. So that's basically how we control the stomach, through either generally through the autonomic system and how the blood gets to the stomach and back out. Now, when we're looking at the stomach microscopic anatomy, right, the stomach wall actually contains 4 tunics or 4 layers. And you can see here, it's the same as what we were talking about earlier when we were looking at the general anatomy of the digestive system, right? The mucosa that has both the epithelium, the lamina propria, and the muscularis mucosa. Then you have the submucosa area that contains the submucosal plexus, right? And then you can see the muscular externa, which contains the myenteric plexus, and you can see all the different layers there in the serosa. I'm not going to go into a bunch of detail here because I've already kind of talked about how this works. The only thing that I'm going to go into detail are just a few of these so that we can tell what's going on. For instance, in the stomach, or the muscularis externa, we have the 3 different layers, right? And so this when we're looking at, we have the circular and the longitudinal smooth muscle layers, but we also have an extra 3rd layer there, right? And so when we're looking at the oblique layer there, right? This is the inner layer there. This allows the stomach to not only churn, mix, and move chyme, but it also allows it to pummel it. So this increases physical breakdown. So that's how this is. The other layers just have these 2 guys, right? Where the stomach has this oblique inner layer that helps. Now, these guys are also a little different when we're looking at the mucosa layer, right? Because this actually makes a 2-layer coat of alkaline mucus. And it makes sense because we are producing in here so much stomach acid to break things down that we want to make sure to protect the lining of the stomach. And so now that we're looking at this, let's look at these. This lining produces all these gastric pits here, right? You can see these openings here, and then this is one of them that we're going to look at. So basically, this is the mucosa layer. And basically, that layer, like I said, is a little modified. It consists of all these basically cells, or this layer that's composed of entirely of these mucus cells. And so you can see all of these mucus cells that are there. Again, if you remember, mucus cells help to produce basically a thin, acidic mucus. Now, they don't really know what its known function is at this point. Now, if you look, these little purple one, or these little turquoise ones right there, are parietal cells. And so they secrete two things. They secrete hydrochloric acid. So the hydrochloric acid is somewhere between pH 1.5 and 3.5. And this has a few things that it does. So let me go over here and write this. And then I'll go back. So this helps to denature proteins. It helps to activate a protein called pepsin, which is going to help to break down proteins. It's going to break down plant cell walls. And it's there to help kill bacteria. Not all, but a lot of them, right? So that's what hydrochloric acid does. Now, to let you know, hydrochloric acid is not the only thing that this parietal cell secretes. It also secretes intrinsic factor. And this is a glycoprotein, and it's required to help with vitamin B12 absorption. And this happens in the small intestines. So we've covered the mucous neck cells, we've covered the parietal cells. Now let's talk about the chief cells. Now the chief cells secrete two things. The first thing they secrete is pepsinogen. And so this is an inactive enzyme that basically gets activated by pepsin. Or it gets activated to, that basically, why does it do that? It's an activated enzyme that gets changed into pepsin due to HCl. So the chief cells secrete pepsinogen. The parietal cells secrete hydrochloric acid. And once pepsinogen comes in contact with hydrochloric acid, it becomes activated into pepsin. It also secretes some lipases. And this digests lipids, about 15% of them. Now you do have the enteroendocrine cells down here at the very bottom, right? And so these guys do a couple things. They secrete chemical messengers. Into the lamina propria, right? So they act as these things called paracrine cells. If you remember in part 1, we talked about these. These act on nearby cells, right? Versus artecrine that acts on the same. Now, they're responsible for secreting serotonin and histamine. So these are a couple of basically hormones that they secrete. In addition to, they also secrete somatostatin and gastrin. And we're going to talk all more about what all these guys do later. So these essentially are these gastric pits open into this area, and these are responsible for producing out of here. These mixtures of cells produce the gastric juices that get released into the stomach. And it's a combination of all these guys working together to produce this, this juice to help with breaking down of helping to break down the food within the stomach. Now this enteroendocrine cell that produces basically this gastrin and the somatostatin, these guys are important because essentially these help to produce a thick layer of bicarbonate rich mucus. Can you see why that might be important? If we're producing all this stuff that's going to basically make the stomach really acidic, which will cause items within the stomach to go through hydrolysis. If we're producing these enzymes that will start tearing away proteins and fats and all this, we want to make sure that we're not actually digesting the cells themselves that are in the stomach. And so here, basically, we want to make sure that everything is working the way that it needs to. So these are going to work together to basically kind of protect or to digest. So the mucosal layer that we have that gets produced protects the stomach and is created by 3 factors basically. Again, a bicarbonate layer, What else also helps us are there are tight junctions in the stomach. It's between the epithelial tissue, so essentially it's going to prevent the juice from seeping underneath the tissue. And basically, mitosis is fast. The surface of this is replacing every 3 to 6 days to help make sure that the damaged epithelial cells are removed quickly. So if anything breaches this mucosal barrier, it can lead to gastritis. And this is the inflammation caused by anything that actually breaches the stomach's mucosal barrier. So things like peptic or gastric ulcers are considered this because they can cause erosions on the stomach wall. And if these erosions actually perforate the wall, it can lead to peritonitis and hemorrhaging, right? And most of these ulcers are caused by Helicobacter pylori, which is a bacteria. And the guy is very interesting that actually hypothesized this. People thought he was full of just nonsense forever. And so what he actually did is he had this idea that he felt like this bacteria caused ulcers. So he infected himself with this bacteria, H. pylori. Then he got ulcers, and then he gave himself an antibiotic to kill this, and his ulcers went away. And then he released and published that data. So it's kind of interesting. They can also be caused by non-steroidal anti-inflammatory drugs, things such as aspirin. And so this kind of shows you the H. pylori bacteria and the mucosal layer of the stomach, and then essentially what an ulcer can look like. And so the digestive process is carried out by the stomach and basically these 6 steps. Basically, it carries out broken down food. It serves as a holding area. It delivers chyme to the small intestines. It denatures proteins. I.e. breaks them down, right, by hydrochloric acid. Pepsin actually carries out protein digestion. Now, casein, which is a milk protein, is a little bit different. It's actually broken down by renin in infants. And so that's why when infants like spit up and it has that curdy kind of substance in milk, that's why it's a little bit different. Now, basically, lipid-soluble alcohol and aspirins are absorbed into the blood through the stomach. And the only stomach function absolutely essential to life is the secretion of intrinsic factor. And if you remember, intrinsic factor is for the absorption of B12, vitamin B12, And if you remember, why is vitamin B12 important? Because it's needed for red blood cell maturation, right? And it can lead to pernicious anemia if you lack this intrinsic factor. So these are all the different digestive processes that are actually carried out by the stomach. So how do we regulate gastric secretion? Well, gastric mucosa secretes about 3 liters of gastric juice per day, and it's regulated by the following. You have neural mechanisms. So basically, the vagus nerve stimulates the increase in secretion, and the sympathetic stimulates the decrease in secretion. You also have, besides neural mechanisms for gastric secretion, you also have hormonal. So gastrin helps to stimulate enzyme release and HCl secretion. So the parietal cells that secrete HCl, gastrin released by the endocrine cells help to increase that. Now, gastrin antagonists are secreted by the small intestines. And this makes sense because if it's starting to get into the small intestines, then you can slow down on the production of enzymes and HCl secreted in the stomach, right? Now, gastrin secretion are broken down into 3 phases. You have the cephalic phase, you have the gastric phase, and the intestinal phase. And so the gastric or cephalic gastric phase for gastric secretion is a conditioned reflex. So think things like aroma, taste, sight, thought. All those first things that happen when you're thinking about eating, right? That's going to be the cephalic or the reflex phase. The gastric phase basically lasts about 3 to 4 hours. This provides about 1/3 of the gastric juices released, right? And so how does this gastric phase get stimulated? Well, basically you get swallowing, distension, or swallowing activates. These stretch receptors. And that's going to initiate both long, the brain, right, and short reflexes, which is your gut reflex. So that's one thing that's going to stimulate it. You can also get chemical stimuli. So things such as partially digested protein, caffeine, low acidity will activate those endocrine cells to secrete gastrin. And basically, once those chemicals, let's say the endocrine cells release gastrin, and basically that release of gastrin then is going to trigger HCl release from the parietal cells, and activate enzyme secretion. So that's stimulation of the gastric phase, right? Now, once we have basically those endocrine cells release not just gastrin, but they also release histamine. And histamine's there to help basically buffer the action of ingested proteins, because those are going to cause a pH to rise, which activates more gastrin. And so this is going to kind of help to buffer some of that action. Now, how can we inhibit the gastric phase? So basically, low pH does. So pH is going to lower between things like between meals, it can happen during eating due to negative feedback. So basically, this may not sound like it makes sense, but think about it. The more protein that you have, the more HCl that's going to be secreted, because HCl is going to, or hydrochloric acid is going to activate pepsinogen into pepsin, which then can break down proteins. Well, that's all fine and good, but it's going to cause a decline in your pH, which will inhibit gastrin secretion over time. So those are two things. Basically, an inhibition of gastric phase is mostly based on low pH, right? Now, the intestinal phase it begins with a brief stimulatory component followed by inhibition. So basically what's going to happen is your food, your partially digested food, is going to enter the intestines, causing a release of enteric or intestinal gastrin. And so this is going to encourage the gastrin glands of the stomach to continue secreting activity. And so basically, let me write this down. So partially digested food, right, goes into small intestines. I'm going to basically, that's small intestine right there. And so this is cause a release of the intestinal otherwise known as enteric gastrin. And so basically you have partially digested food, maybe not all the way, it gets into the small intestine, so it's going to tell the stomach, hey, release it, release more gastrin, gastric juices into the stomach so we can continue secreting the activity and breaking things down. Now, when do we stop this? When in the intestinal phase, what inhibits this? There's four main factors in the duodenum, that area that it hits right after it gets out of the stomach, that causes inhibition of gastric secretions. The first is the distension of the duodenum. So this is the basically the stretching. And this is due to, why is this so, can't really, sorry. Oh no. Of duodenum, and this is due to the chyme actually entering it. Another thing is the presence of acidic chyme, fatty chyme, or hypertonic chyme. So these inhibitory effects actually protect the intestines from being overwhelmed by too much chyme or too much acidity that it can't actually take, right? The inhibition is achieved in a couple different ways. And that's what we're going to talk about next on our next slide. So it's inhibited by the enterogastric reflex and the enterogastrons. So the enterogastric reflex is in the duodenum. It actually inhibits acid secretion in the stomach by interacting with the enteric nervous system, which is the short reflex, and the sympathetic nervous system and vagus nerve, which is the long reflex. So basically gut-brain, brain-brain, right? And it also works with the duodenal enteroendocrine cells, which release two important hormones that inhibit gastric secretion, and that's secretin and CCK, or cholecytokinin. So let's see what both of these guys do. Just to let you know, the basically how all this intestinal phase works is that the stimulatory effects is very brief. So when I'm talking about stimulation happening here, it's very, very short time. It's towards the beginning as stuff is starting to enter in the duodenum. But it's overridden by the inhibitory stimulation as the intestine starts filling, right? And so here is a CCK, and you can see hormones in the paracrine that act in, and we talk a lot about different ones, but this is used to help basically with the inhibition of some of the gastrin released during the intestinal phase. And so the site of production is in the duodenum, in the mucosa of the duodenum. It's basically fatty chyme and partially digested proteins that help with the stimulus for production. And basically, these are all the different areas for which it'll target. And basically, it helps the main part of this that we're going, it inhibits stomach secretory activity. So it basically helps to reduce the gastric juices that are being produced in the stomach once we get into the intestinal phase. And the other one is the secretin. Again, you see it made in the mucosa of the duodenum, and its stimulation is partially digested proteins and fats, again, basically acidic chyme, and its target is the stomach, pancreas, and liver. And in here, as you can see again, it inhibits the gastric gland secretion and gastric motility, right? And so these are the two things. Of course, there's other things in here that they're talking about, but those are the two main that we're focusing on in the looking at the inhibition of the intestinal phase. So let's look at the neurohormonal mechanisms that regulate or stimulate or inhibit gastric juices. So this is just a review of what we just talked about. So the cephalic phase, basically when you're looking at this site and thought of food, goes to the cerebral cortex, through the hypothalamus and the medulla oblongata, through the vagus nerve, into the tummy, and basically this is going to start with gastric juice production. Add to that also taste and smell receptors communicate, and all of this goes down and starts producing basically gastric juices. Now, and once that happens, you can see on here the gastric phase. Your stomach actually starts to stretch, that distension. I think I said swallow earlier, but I meant to say stretch. It activates those stretch receptors, and basically your long reflex, which is through the medulla and vagus nerve again, so we're looking at the brain, will go in there and actually activate gastric juice production. And then if you look, the short reflex, which is the gut brain, will come in there and activate that as well. The second thing that's important on there are food chemicals like peptides and caffeine. They're going to help raise the pH, which will activate chemoreceptors, activate the G cells, and the G cells are those enteric endocrine cells that will release gastrin to the blood, and that will activate gastric acids in the stomach, right? And then once you get through the tummy area and you're going into the duodenum, we'll get into the intestinal phase. And basically here, once you get a small amount of partially digested food into that area of the duodenum, or distension of the duodenum, a bit of stretching, when the stomach begins to empty into there, you'll get a brief effect where it will do the intestinal enteric release of gastrin to the blood. This is brief though, right? Immediately after that, you get into the basically the inhibitory effects. And basically with here, once you get a certain amount of stretching of the duodenum, a presence of fatty, acidic, or hypertonic chyme, then you're going to get a inhibitory effect. So you're going to get secretin in that CKK, cholecystokinin, or CKK, that gets released, and that's going to basically, these are entogastrons that will reduce the secretion, or the intergastric reflex, which both includes the both short and long reflexes, both gut brain and regular brain, to stop. Now, when we're talking about the inhibitory of the gastric phases, basically if it becomes too low, or the pH is too low, then essentially what you can see is the G cells will basically reduce or decrease the amount of gastrin released to the blood, and so that will reduce the amount of gastric juices secreted. Also, emotional stress can activate the sympathetic nervous system and cause you to override the parasympathetic and reduce gastric juices. And to go with emotional stress, loss of appetite and depression can also cause a decrease in parasympathetic activity and inhibit. So how is HCl secreted by the parietal cells to begin with? Because this is a big part. About how things are secreted. Give me 1 second. So, if you can see, we're taking a bit out here. Of HCl and what happens. So here's the blood capillaries, the interstitial space right here, and the actual gastric gland. And so if you look at the first, we're going to have both bicarbonate and HCl are generated from the dissociation of carbonic acid produced from CO2 and H2O. And so if you look here, you have water that's available here, and you have carbon dioxide coming in. So you have all of this carbon dioxide, basically all of these different things happening or coming into the blood, which comes over into this interstitial fluid. And so the CO2 is going to combine with the water to make carbonic acid, which is right here, right? And basically the carbonic acid is going to break down into basically an H and the bicarbonate. From here, this hydrogen or proton is going to be pumped using the hydrogen potassium ATP pump into the lumen of the stomach. Of course, the K, or the potassium, returns to the lumen through the membrane channel. So you can see on here, it's going here and then it's coming back, right? The main point of this is to try to get the H on the other side. Now, this guy, the next part that we're looking at is within the interstitial fluid, you also have calcium. And so on here, we're getting calcium to come in and bicarbonate to leave, because that's how this pump works. So you're getting basically bicarbonate going out to help buffer the blood. And then from that, we're able to get chlorine in. And so this is going to diffuse into here and basically make HCl. So when we're talking about how we're getting HCl made by these cells, right? What you can do is the H is coming from the breakdown of carbonic acid. That's coming from water and CO2, right? And the Cl is coming from basically the blood and the interstitial fluid, and it's an exchange between the bicarbonate and the Cl, right? So that's how it's working. And that little cell is doing all that work just to get hydrochloric acid. So, when we're talking about gastric filling, or how we regulate gastric filling, it's a response of the stomach to filling. So there's basically stretch receptors there, right? And so in your stomach, you can keep bringing food in because you're able to accommodate the stretching, right? And there are two factors that cause pressure to remain constant until we get 1.5 liters of food is ingested. So you have receptive relaxation and you have gastric accommodation. And so receptive relaxation is the idea that the stomach, you can relax the stomach due to swallowing. So as you swallow, it's going to relax to cause the stomach to be able to fill. And basically, this is a coordinated effort using the swallowing centers. In the brainstem. So the gastric accommodation is this idea that it stretches to decrease tension and then it relaxes. It's kind of the idea that if you're stretching for the splits, you stretch for a while, then you relax, and then you can stretch longer the next time, right? So this is what's known as a stress relaxation response. And this is what allows the hollow organs to stretch without increasing tension or contractions. So that's how you're able to get more food while you feel. How do we allow the motility and the emptying of the food once it gets in there? Well, you have gastric contractile activity. So you have this peristaltic wave that moves towards the pylorus at a rate of 3 per minute. You have what's called a basic electrical rhythm, or BER, is set by what's known as these things called enteric pacemaker cells. Think of the same idea as the heart, that the heart could basically contract on its own because it had its own pacemaker cells. So the stomach does as well. They're linked by gap junctions, so the entire muscular contracts together. Distension and gastrin increase the force of contraction. So the more it stretches and the more gastrin that gets released, the more it increases the force of the contraction to try to empty it. Contractions are most vigorous and powerful close to the pylorus, or that region where it's the pyloric sphincter where it's emptying, right? And 30 milliliters of chyme is produced. And so when it's delivered to the duodenum, it's delivered in about 3 milliliter spurts. And then the rest of it, the 27 other mil, is forced back. And this little valve, this pyloric valve that you're going from the stomach to the duodenum, only liquids and very small particles are allowed to pass through it. And so when you're looking at this, the peristaltic waves in the stomach, how we move, you can see it contracts here, and so you have propulsion. That's this peristaltic move. Waves move from the fundus up here down to the pylorus, which is here, right? And so you can see that it's starting to move down. And basically, then you have grinding. And so that's the most vigorous peristalsis. And mixing action occurs closest to here, the pylorus, right? The pyloric end of the stomach acts as a pump that delivers small amounts of chyme into the duodenum. Remember, there's 30 mils, but only 3 mils go at a time. And so that bunch of that doesn't get to come out gets forced back. So it's called retropulsion. And the peristaltic wave closes the pyloric valve here, forcing the contents of the pylorus back into the stomach. So that's going to essentially help to mix that stuff better as well. So as we're emptying the stomach, how does the duodenum prevent from overfilling? And basically, they prevent from overfilling by controlling chyme entry. And so if you remember what we were just talking about, you have in here duodenal receptors in that area, in the duodenum, right? That respond to stretch and chemical signals. You also have both the reflexes, remember? The enterogastric reflex and enterogastrons. Secretin in the CKK that's there that inhibit basically gastrin or gastric secretions, right? And the filling. So the stomach will empty in about 4 hours, but increase in fatty chyme entering duodenum can increase time to about 6 hours or more. Right? And then you have the carbohydrate-rich chyme moves quickly through the duodenum. And we are so close to the end, guys. Just hold on just a few more. So on this emptying factor, right, the neural and hormonal factors that inhibit gastric emptying, again, any kind of presence of chyme that's fatty, hypertonic, or acidic, right, that's going to come and that's going to basically going to be picked up by the chemoreceptors and the stretch receptors, and also the duodenal enteroendocrine cells. They're going to secrete the CKK or the secretin, and that's going to act and reduce contractile force on the stomach and reduce the rate of stomach emptying, aka the amount of gastrin produced as well. On here, those chemoreceptors and stretch receptors will activate or trigger the enterogastric reflex. The short reflex enters through the enteric nerve. So this is the gut brain, right? The long reflex is through the central nervous system, will increase sympathetic, decrease parasympathetic, and basically will cause the reduction in contractile force. Now, once this happens, this can actually come and inhibit this presence after a time. So, and this, you know, we've just talked. So this is the enterogastrons that can be secreted that'll affect, and this is the enterogastric reflex. I think I'm almost killing that to the point of being ridiculous. So homeostatic imbalances that can arise from the stomach. Vomiting is basically caused by extreme stretching. And so this is intestinal, right? Extreme stretching can cause this. Intestinal irritants such as bacterial toxins or excessive alcohol, spicy food, certain drugs can all cause vomiting. And basically what happens is chemical and sensory impulses stimulate the emmeric center in the medulla. So the problem with excessive vomiting, it can lead to dehydration and electrolyte and acid base. And so alkalosis, right? And so a lot of times we want to get that under control. So now let's go, and we've basically beat the stomach to death. Let's go and talk about some of the other things that work. So the liver, the gallbladder, and pancreas are accessory organs. They're not actually part of the tubing that goes through. So the liver is important because it has the digestive function, and a main part of it is for the production of bile. And bile is a fat emulsor. So it's not so much going to digest fat, but it's going to allow big clumps of fat to be put into smaller kind of droplets. The gallbladder, its chief function is the storage of bile. And the pancreas supplies most of the enzymes to digest chyme. It also is important because it produces most of the bicarbonate to neutralize stomach acid.
There's a factory inside you that weighs about 1.4 kilograms and runs for 24 hours a day. This is your liver, the heaviest organ in your body, and one of the most crucial. This industrious structure simultaneously acts as a storehouse, a manufacturing hub, and a processing plant. And each of these functions involves so many important subtasks that without the liver, our bodies would simply stop working. One of the liver's main functions is to filter the body's blood, which it receives in regular shipments from two sources. The hepatic artery delivers blood from the heart, while the hepatic portal vein brings it from the intestine. This double delivery fills the liver with nutrients that it then sorts, processes, and stores with the help of thousands of tiny internal processing plants known as lobules. Both blood flows also deliver the oxygen that the liver needs to function. The blood that is received from the intestine contains carbohydrates, fats, and vitamins and other nutrients dissolved in it from the food you've consumed. These must be processed in different ways. In the case of carbohydrates, the liver breaks them down and converts them into sugars for the body to use as energy when the filtered blood is sent back out. Sometimes the body has leftovers of nutrients that it doesn't immediately require. When that happens, the liver holds some back and stacks them in its storage facility. This facility works like a pantry for future cases when the body might be in need of nutrients. But the blood flowing into the liver isn't always full of good things. It also contains toxins and byproducts that the body can't use, and the liver monitors these strictly. When it spots a useless or toxic substance, it either converts it into a product that can't hurt the body, or isolates it and whisks it away, channeling it through the kidneys and intestine to be excreted. Of course, we wouldn't consider the liver a factory if it didn't also manufacture things. This organ makes everything from various blood plasma proteins that transport fatty acids and help form blood clots, to the cholesterol that helps the body create hormones. It also makes vitamin D and substances that help digestion. But one of its most vital products is bile. Like an eco-friendly treatment plant, the liver uses cells called hepatocytes to convert toxic waste products into this bitter greenish liquid. As it's produced, bile is funneled into a small container below the liver, called the gallbladder, before being trickled into the intestine to help break down fats, destroy microbes, and neutralize extra stomach acid. Bile also helps carry other toxins and byproducts from the liver out of the body. So as you can see, the liver is an extremely efficient industrial site, performing multiple tasks that support each other. But such a complex system needs to be kept running smoothly by keeping it healthy and not overloading it with more toxins than it can handle. This is one factory we simply can't afford to shut down.
So let's look at the gross anatomy of the liver really quick, and we're going to do this in class as well, right? This is the largest gland in the body. It weighs something about 3 pounds. Right? It consists of 4 lobes. You have the right lobe here that you can see very easily, and that is separated here. Actually, let me do, oops. This whole area down here is the right lobe. Then you can see the left lobe over here, and the two is separated by this falciform ligament that's in the middle. That's going to separate the right and the left lobes. Also, it suspends the liver from the diaphragm and the anterior abdominal wall. Now, if we're looking for the other lobes, you're going to have to flip around it, and you'll see that you can see here. Here's the left lobe. Again, this is the right lobe over here, and you can see on the bottom here towards the back, you can see this part right here, which is the caudate lobe. And then the lobe that's between this falciform ligament and the gallbladder at the bottom is the quadrate lobe, right? Now, if you look here, you see the filiform ligament, and then a little farther, you see this round ligament. This is also known as the ligamentum teres. And this is the remnant of the fetal umbilical vein. Umbilical vein. So that's along the free edge of the falciform ligament. Let's see. You can see here you have the gallbladder, which is going to hold the bile. You have from there, you have the cystic duct that is connecting to the gallbladder. You can see this common hepatic duct. This is the duct that leaves the liver. And basically the bile duct, which you can see right here, is the connection of the cystic duct and the common hepatic duct together. Let me see, let me go back one. And so you can see here, okay. You can see on here as well, you have the hepatic veins here, and you have the hepatic artery here, and they basically enter the liver at this porta hepatis region. Now, when we look at the liver itself, this is a large anatomy. We can look at the microscopic anatomy, and when we look, this is the anatomy of the liver itself. And so on here, you can see we're taking the liver and we're taking these, lobules right here, and we're blowing them up, right? And so these guys are composed of basically hepatocytes. And so you can see that this lobule up here, right, you can see all those. So these little individual things here are all hepatocytes. And those cells help to filter and process nutrient-rich blood. You can see the central vein here. And that's located in this longitudinal axis. If you look in each corner of the lobe, it'll contain what's known as a portal triad. And the portal triad is made up of three things: The portal triad is made up of this, and you can see where the green dots are, the hepatic artery. This supplies O2. The hepatic portal vein, which brings the nutrient-rich blood from the intestines. And the bile duct up here, which is going to essentially receive bile. And it receives bile from these little guys right here, if you can see. It's the bile caniculus. Now you also have these sinusoids that you can see here. These are leaky capillaries, and they're located between the hepatic plates. And so the blood from both the hepatic portal vein and the hepatic artery actually percolate through the triad region that I just showed you here, right? And from the triad region, they go through the sinusoid and empty into this central vein area here. And you can see on every single one of these, they have these portal, these triads. And you can see they're all moving into the middle here. So you can see again, just to review them, this whole thing, right, is the lobules. They're composed again of hepatic cells or hepatocytes. They help to filter and process nutrient-rich blood. You can see this central vein here. And so what happens, you have these triads that are in the corner, right? And if you remember, these arteries supply O2. The portal veins bring nutrient-rich blood from the intestines. The bile ducts receive bile. And then all of that is going to go and move through these sinusoids to empty into the central vein. Now, these bile caniculi here are going to be what ends up bringing the bile into the bile duct. And within this area, you have stellate macrophages, otherwise also known as hepatic. And these are in the liver sinusoids. And they help to remove debris and old red blood cells. So let's really quick talk about the hepatocytes. And so the hepatocytes have increased rough and smooth ER, Golgi apparatus, peroxisomes, and mitochondria. And they have the following functions. They help to produce about 900 milliliters of bile a day. They process bloodborne nutrients. Like example, store glucose as glycogen and make plasma proteins. They store fat-soluble vitamins. And they detoxify. So, perfect example, they take something like ammonia and they convert it into urea. Now, when we're talking about hepatocytes making bile, what exactly is bile? And what it is actually this yellow-green alkaline solution, and it contains bile salts, bilirubin, and things like cholesterol, triglycerides, phospholipids, and electrolytes. And basically, the enterohepatic circulation helps to recycle bile to conserve bile salts. And so there are some homeostatic imbalances of the liver. Hepatitis, for instance. This is usually a viral infection or can happen due to drug toxicity or wild mushroom poisoning, or something like cirrhosis, which is a progressive chronic inflammation from chronic hepatitis or alcoholism. So basically what happens is the liver, those areas get converted into fatty, fibrous, useless cells, and this leads to what's known as portal hypertension. And so basically the liver isn't able to detoxify your body the way it should. And so a lot of times liver transplants are successful, but the problem is livers are scarce. Liver can regenerate to its full size in 6 to 12 months after 80% of it is removed, but you have to have some of it left, and that part needs to be somewhat healthy. So it can cause a bit of a problem. Now the gallbladder. This guy is a thin-walled muscular sac. It's on the ventral surface of the liver. And its function is to store and concentrate bile. And they do that by absorbing water and ions. It contains many honeycomb folds that allow it to expand as it fills. And basically, muscular contractions release bile via the cystic duct, which flows into the bile duct. You can get basically gallstones is a homeostatic imbalance. It's caused by too much cholesterol or too few bile salts. It can cause obstructions of flow of bile from the gallbladder. It's painful when the gallbladder contracts against sharp objects, crystallized objects. So this can be very painful. Obstructive jaundice is also an issue that can happen, and this is a blockage that causes bile salts and pigments to build up in the blood, resulting in jaundice or yellow skin. Jaundice can also be caused by liver failure. And basically, gallstone treatment is taking either crystalline dissolved drugs, ultrasound vibrations can allow movement, laser vaporization, or surgery. And now we're going to hit on what the pancreas is.
Beneath your ribs, you'll find, among other things, the pancreas, an organ that works a lot like a personal health coach. This organ controls your sugar levels and produces a special juice that releases the nutrients from your food to help keep you in the best possible shape. The pancreas sits just behind your stomach, an appropriate home, as one of its jobs is to break down the food you eat. It aids digestion by producing a special tonic made of water, sodium bicarbonate, and digestive enzymes. Sodium bicarbonate neutralizes the stomach's natural acidity, so these digestive enzymes can perform their jobs. Lipase breaks down fatty substances. Protease splits up proteins. And amylase divides carbohydrates to create energy-rich sugars. Most of those nutrients then get absorbed into the bloodstream and go on to enrich the body. While all this is happening, the pancreas works on another critical task, controlling the amount of sugar in your blood. It achieves this with the hormones insulin and glucagon, which are produced in special cells called the islets of Langerhans. Having too much or too little sugar can be life-threatening, so the pancreas must stay on constant alert. After a big meal, the blood often becomes flushed with sugar. To bring us back to normal, the pancreas releases insulin, which makes the excess sugar move into cells where it's either used as an energy source or stored for later. Insulin also tells the liver to shut down sugar production. On the other hand, if blood sugar is low, the pancreas releases a hormone called glucagon that tells the body's cells and liver to release stored sugars back into the bloodstream. The interplay between insulin and glucagon is what keeps our sugar levels balanced. But a faulty pancreas can no longer coach us like this, meaning that this healthy balance is destroyed. If it's weakened by disease, the organ's ability to produce insulin may be reduced or even extinguished, which can trigger the condition known as diabetes. Without regular insulin release, sugar steadily builds up in the blood, eventually hardening the blood vessels and causing heart attacks, kidney failure, and strokes. The same lack of insulin deprives cells of the energy-rich sugar they need to grow and function. People with diabetes also tend to have higher levels of glucagon, which makes even more sugar circulate. Without this internal health coach, our sugar levels would go haywire, and we wouldn't be able to digest important nutrients. But like any coach, it's not the pancreas's job alone to keep us healthy. It needs our conscious participation too.
So I hope that helps a little bit. So let's talk about the pancreas. It has exocrine and endocrine function. So if you're looking at the cell here, you'll see a small duct, and you can see basically you have the acne, which is a cluster of secretory cells. They produce zymogen granules. Which actually contain what's known as proenzyme. Now you can see on there that you also have ducts. And so these guys are going to secrete to the duodenum via the main pancreatic duct. Smaller duct cells can produce water and bicarbonate. So you can see the center cells that are there to help basically secrete enzymes, and you can see these zymogen granules that are in there that are basically the proenzymes before they're actually activated. And then you can see that that is incorporated in one of these acinas, and then the small ducts go into here, and these are going to actually secrete to the duodenum via the pancreatic duct. So this is the exocrine function. Now they do have an endocrine function because it secretes both insulin and glucagon. And these are made by the pancreatic islet cells. And so you can see these small duct cells that create the bicarbonate and water are there as well. And these are the larger ones that help to secrete the actual enzymes that we're going to use to digest. And so about 1,200 to 1,500 mils per day of pancreatic juices are produced. It's a watery alkaline solution, about a pH of 8. And essentially, it's there to neutralize the acid chyme coming from the stomach. Wait a second, I'm trying to figure out. And so basically, you have the electrolytes are produced primarily, but then you also have digestive enzymes. And so the digestive enzymes that we have are things like proteases. And so these are for proteins. And they are released in an inactive form. You don't want to have self-digestion. You have amylases, which are for carbs. You have lipases, which are for lipids. And then you have nucleases, which are for nucleic acids. And so the pancreatic juices, the proteases are secreted in an active form. They are activated after they reach the duodenum. And basically they're activated by this enteropeptidase, right? And so this guy is going to interact or activate trypsinogen to trypsin. And once trypsin is activated, it can then activate more trypsinogen. And then you also have things, like procarboxypeptidase. Gets activated into, you just take the pro into carboxypeptidase, and then you have basically chymotrypsinogen that gets activated too. Basically chymotrypsin. And you can see them all over here. Realize that they were over there. And so these are all the inactive forms, and basically you can see as trypsin gets activated, it helps to activate these other. And so these are all examples of proteases. They're all going to break down different proteins or types of proteins. And so you can see how everything's kind of tied together here, right? You have the liver here, and you can see that you have the hepatic ducts here and here. Here's your gallbladder, and then they all go into, and you can see the bile duct here. That kind of goes, and it all kind of combines over here together, where you have this main pancreatic duct and sphincter head together and release here in this major duodenal papilla area here of the duodenum. And so the bulb-like area is what's known as the hepatopancreatic ampulla, and what closes it and opens it is the sphincter. And so this guy controls how much bile and pancreatic juices get in there. And this accessory duct that's right here dumps directly into the duodenum. It's much smaller. And so, mechanisms that promote secretion and release of bile and pancreatic juices. So bile and the pancreatic juice secretions are both stimulated by neuronal and hormonal controls. And so the hormonal controls are just like we were talking about earlier. Oops, sorry guys. Wait. You have the CCK and secretin that are secreted by the duodenal enteroendocrine cells. And so the CCK is stimulated by protein and fat in the chyme. The secretin is released due to acidic chyme. And basically, these two guys enter the blood circulation and cause the following events to happen. First, it causes pancreatic secretion, right? And so you can see this is going into the pancreas. And so the CCK is going to induces secretion by the center cells of the enzymatic pancreatic juices. And the secretin causes secretion by duct cells of the bicarbonate-rich pancreatic juices. And so the vagus nerve is weakly stimulated during the cephalic and gastric phase to cause change here too. Now from here, you can see that the CCK and secretin activates the liver to start secreting bile. And the bile salts returned from the interhepatic circulation are the most powerful stimulus for bile secretion. Secretin is such a minor stimulus that it's not as important. So here, it's all about the bile salts, right? After that, gallbladder contracts, because that's what's holding the bile, right? And CCK has it to contract. The vagus nerve does have some workings in this, but it's really weak. And so you can see here the hepatopancreatic sphincter is going to relax. And CCK causes that sphincter to relax. And so at that point, bile and pancreatic juice actually enters the duodenum. So CCK and secretin are important throughout this. Both of them are important in this first part when we're talking about the pancreatic secretions actually happening. Once that happens, basically bile, you'll see some of this CCK coming into the liver is more important here. But for bile secretion, the most powerful part of it is the returning of bile salts, right? And then you'll see the gallbladder contracts to release it due to CCK. And then basically the sphincter will relax because of CCK. So the only time secretin is a really big factor is the very first part when we're talking about the cells actually secreting it to talk to the pancreas. So secretin's really important in pancreatic juices, and the rest of the area it's not so much. And the small intestines length and other structural modifications provide huge surface area for nutrient absorption. So that's what's important here. And so there are some modifications. And so if you look at here, you'll notice on there that you have these microvilli that are part of this brush border cells that help with surface areas. So you can see there's so much more area that's exposed. So that's going to help the basically the absorption of nutrients. Missing. And so when we're talking about the digestive process in the small intestines, the chyme from the stomach actually contains partially digested carbohydrates and proteins and undigested fats. Hold on here really quick. And so when we're talking about how long it takes the small intestine to absorb all the nutrients and most of the water, on average, 50% of the stomach content empties about 2.5 to 3 hours and completely empties about 4 to 5 hours. So it requires about 2.5 hours to 3 hours before 50% of the content in the small intestines actually emptied. And at this point, your digestive system has absorbed most of the nutrients, right? Sorry. So sources of sources of enzyme for digestion, substances such as bile, bicarbonates, digestive enzymes, not brush border enzymes, are imported from the liver and pancreas. Brush border enzymes found in the plasma membrane perform final digestion of chyme. And so the process in the small intestines, how it moves, after a meal is segmentation to break everything up, but between meals, it's peristalsis to make sure to get rid of everything. And then the last part of this is the large intestines, and this is the area where we're just going to basically absorb water and get rid of what we've digested. And you can see in here, it's made up of 3 parts: the ascending, transverse, and descending colon, along with the sigmoid colon at the end. Towards the bottom, you'll get the rectum and the anal canal with both external anal sphincter and an internal anal sphincter. You can see the mesenteric here as well. And of course, the very first, as you can see, this is the ileum. That comes from the last part. So you have the duodenum, the jejunum, and the ileum of the small intestines. And so the ileum comes out to enter this first areum called the cecum into the small large intestines. And then it'll start coming up from there. Of course, the appendix, where some people, it's still kind of unknown completely what it does. It aids in some digestion and to catch some things down there that aren't as digestible. And the big help of the large intestines is really just to get rid of water and to kind of compact. And since this was such a long lecture talking about other things, we're not going to go much into the process of the large intestines. So we are